Welcome back to Dead Reckoning, the Human Life Expectancy channel. Having resolved the mystery of why we age in our last video, in this video we'll discuss the hallmarks of aging and what makes us think that aging might actually be cured. Based on decades of studies of humans and animals of varying lifespans, the science of aging has advanced rapidly. From a universe of possibilities, scientists have now significantly narrowed the potential biological drivers of aging. A recent paper titled The Hallmarks of Aging by Carlos Lopez-Ontin and collaborators builds upon the prior work of Aubrey de Grey and others to highlight 9 factors that seem to drive mammalian aging. Each of these hallmarks has been scientifically proven to meet 3 conditions. First, it increases with age. Second, accelerating the hallmark's progress increases aging. And third, reducing a hallmark's progress reduces aging. The 9 hallmarks are genetic instability, telomere attrition, epigenetic alterations, loss of proteostasis, deregulated nutrient sensing, mitochondrial dysfunction, cellular senescence, stem cell exhaustion, and altered intercellular communication. To provide the background for how each of the technologies that we're going to discuss might impact aging itself, let's briefly describe each of these hallmarks. Genetic instability results when damage from metabolism, viruses, radiation, pollution, or other factors causes modifications to our DNA. One study estimated that an individual cell can suffer up to 1 million DNA changes per day. Fortunately, DNA has natural processes to identify and fix this damage, but the repair is imperfect and the damage does accumulate with age. This can cause cells to become cancerous, senescent, releasing harmful chemicals, and even to die. Since a recent study found that damaged DNA is primarily responsible for aging phenotypes, genetic instability is clearly an important hallmark of aging. Telomere attrition is damage to and the reduction in the length of the tips of DNA molecules that often occurs as a result of repetitive cell division. Often compared to aglets at the ends of shoelaces, when telomeres are damaged, DNA can unravel and have problems replicating. The enzyme telomerase can protect and repair telomeres, but too much can cause cells to become cancerous. The next hallmark of aging is epigenetic alterations. As you age, not only do your genes change, but the way your genes are expressed changes. In effect, as cells age, they can lose their memories of what type of cell they should be and how they should behave, causing a host of problems in how they function and coordinate with other cells. Loss of proteostasis occurs when proteins age and lose their ability to maintain their original forms and abundance. Healthy protein lifetimes can vary from minutes to years, after which they're degraded and replaced by newly synthesized protein. However, over time, proteins can become misfolded or damaged or clumped together, as in the case of beta-amyloid plaques in the Alzheimer's disease, interfering with healthy protein turnover and their ability to function. Since proteins provide cell structure and regulate virtually all of the chemical reactions in our bodies, this also causes a host of problems as we age. Deregulated nutrient sensing occurs as we age, and our cells become less responsive to whether nutrients are present or not. Healthy cells usually grow and reproduce when nutrients are available, and focus on maintenance and repair when nutrients are scarce. They also tend to grow more when we're young and focus more on repair as we age. Today, many of us now spend most or all of our lives with excess nutrients available from overeating, causing diseases such as cancer that result from insufficient maintenance and repair. More broadly, as we age, our cells become less responsive to nutrients, growing in their absence or failing to grow in their presence, again causing increasing dysfunction throughout our bodies. The mitochondrial dysfunction hallmark of aging is the reduction of our cells' ability to produce energy as we age. Common cells contain many mitochondria that are responsible for respiration and energy production. As cells age, mitochondria tend to be fewer in number and less efficient than those in younger cells. Mitochondria can also produce most of the oxidative damage from free radicals, or as scientists call them, reactive oxygen species, ROS, in your cells. Maintaining the right level of ROS is important because too much kills too many cells, but too little can lead to insufficient response to stress, cancer, and other conditions. As we age, this balance is often upset. Cellular senescence occurs when cells that once replicated vigorously enter a permanent, non-dividing state and secrete damaging molecules into the surrounding area. In the natural process called autophagy, which means self-eating in Latin, senescent cells are consumed. However, when this process is disrupted, as often occurs with age, damaged cells and proteins accumulate, causing a range of health problems. Stem cell exhaustion occurs when stem cells, which create new cells in virtually all of our tissues and organs, die as we age, impairing the ability of our tissues and organs to regenerate and repair damage. Again, balance is key. Healthy stem cells must replicate when required, but not otherwise. As we age, this balance is often upset, causing there to be too few of the cells we need and/or too many of the cells we do not. And finally, the last proven hallmark of aging is altered intercellular communication. Altered intercellular communication occurs when our cells trigger dysfunctional changes in other cells, causing inflammation or other failures. Cells and tissues must communicate for us to remain healthy. For example, hormones produced in the brain alter the way cells behave in the rest of the body. Your liver might chemically tell your brain to produce hormone production, and nerve cells that signal pain can alert your immune system in the rest of your body. As we age, low-level chronic inflammation can become damaging to surrounding tissue, interfering with this communication. While each of these hallmarks has been proven as a separate mechanism of aging, another recent study shows that they're also interlinked. This is good news because it suggests that making progress on some of the factors could have positive effects on the others, advancing our ability to cure aging. We'll refer back to these hallmarks of aging as we present new life-extending technologies in upcoming videos. So, what makes us think that we can cure aging? Based on our advancing understanding of the hallmarks of aging, there are now many good reasons to believe that we can cure aging. First, as we noted in our last video, animals naturally have very different life expectancies, and some appear even not to age at all. Understanding the drivers of this difference uncovers numerous opportunities to extend life expectancy. Second, we've already materially extended the life expectancies of multiple other species. This gives us confidence that similar results can be achieved for humans. The life expectancy for worms, for example, C. elegans, has been extended by 10 times in a study by Cynthia Kenyon at UCSF, which used genetic engineering to change a single letter in their DNA. These worms also remained active and vivacious as their lives were extended. Other studies have shown that the life expectancies of yeast, flies, mice, and rats can be extended between 20 and 40%. Significant extensions for dogs and primates have also been shown. Numerous studies also show that age-associated epigenetic signatures of other species are slowed by various medical interventions. We'll discuss the new technological and medical advances that cause these life extensions in future videos. The third thing that makes us think we can cure aging are the numerous studies showing that changing our behaviors can help us to live longer. Together, eating and sleeping well, exercising, quitting smoking and excessive drinking, and other behaviors have been proven to extend our lives by a decade or more. Fourth, actuaries, biologists, and great thinkers throughout history have said that the human lifespan may not be limited. As we mentioned in our last video, in 1825, Benjamin Gompertz observed that while human risk of death increases exponentially throughout middle age, as people enter old age, the risk of death plateaus. He wrote, quote, "The limit to the possible duration of life is a subject not likely ever to be determined, even should it exist." Even more directly, physicist Richard Feynman noted in 1964, quote, "There's nothing in biology yet found that indicates the inevitability of death." Elisabetta Barbi and collaborators published a study in 2018 that, quote, "strongly suggests that longevity is continuing to increase over time and that a limit, if any, has not yet been reached." In a more recent talk at Google, David Sinclair envisioned a future in which people receive similar treatments every decade or so to undo the effects of aging throughout their body. Quote, "We don't know how many times you can reset," he said. "It might be 3, it might be 3,000. And if you can reset your body 3,000 times, then things get really interesting. I don't know if any of you want to live for 1,000 years, but I also don't know if it's going to be possible. But these are the questions we'll have to start thinking about, because it's not a question of if, it's now a question of when." Fifth, there's good reason to think that biological clocks can be reset. In a very real sense, the aging problem has already been solved. Every time we have children, 2 older people create a new being of age 0. This means that our cells already have an innate ability to reset themselves. The astonishing fact is that germ cells, our gametes, which consist of eggs and sperm cells, have already evolved to reset their biological clocks to 0. If this were not possible, then our children would be born old, which of course they are not. Also, as we covered in our Revolutionizing Human Reproduction video, in 2006 we got scientific proof that human cells could be rejuvenated. Shinya Yamanaka and his team discovered that proteins encoded by 4 master genes could transform any adult cell back into an induced pluripotent stem cell, an iPSC, from which all cells grow. Yamanaka and his team shared the Nobel Prize in 2012 for this contribution. Scientists have also reprogrammed these iPSCs into egg cells for sheep and other mammals and used them to produce normal adults for those species. A 2013 study documented the success in repeated cloning of 25 generations with no increase in difficulty and no reduction in lifespans. Sixth, cancer and other types of cells can effectively live forever. Since these cells continually renew themselves, in principle, there's no reason why an organism, which after all is just a group of cells, could not continue to exist forever. For example, the cancer cells that were taken from Henrietta Lacks in 1951 are still reproducing. Today, these incredible cells, nicknamed HeLa cells, are used to identify the effects of toxins, drugs, hormones, and viruses on the growth of cancer cells without experimenting on humans. They've been used to test the effects of radiations and poisons, to study the human genome, to learn more about how viruses work, and played a crucial role in development of the polio vaccine. Even more, since every cell is the result of a previously living and dividing cell, the cells that are in your body right now are the direct descendants in a continuous line of living cells dating back to the first single-celled organism that evolved 3.8 billion years ago. So, how old are you really? The continuous process of evolution, which we are all engaged in, is extraordinarily robust and timeless. These and other astounding facts and discoveries give us strong reasons to think that aging can be cured. Understanding the hallmarks of aging illuminates what might be possible. This is much more promise than should be necessary to qualify the study of aging as a top priority for doctors, technologists, and for human beings. For all these reasons, in July 2018, a joint proposal to classify aging as a disease was submitted to the World Health Organization's ICD-11 task force by researchers of the Biogerontology Research Foundation and the International Longevity Alliance, and it resulted in the implementation of an extension code for age-related. This was big news because it opened the door for funding earmarked for curing diseases to flow to the study of aging and allows doctors to begin prescribing medications to treat aging. With this background on aging, our next videos focus on new technologies that are showing promise in being able to extend human life expectancy by curing aging. In the meantime, please hit the like button below, subscribe to the Human Life Expectancy channel to make sure you don't miss our next videos, and post comments for the topics you'd like to have us cover. Thanks for joining us. Till next time, as we say at Human Life Expectancy Inc., Hi Hua! Please, do what you can to help yourself and help us all to live longer.